Good morning, students. This is your dedicated Shavika Goyal. Welcome to the class. How are you all? Okay, I hope you all are good. So, students, for today's our topic is meiotic cell division and its significance. Now, first of all, learn what is meiosis. So, meiosis is basically a type of cell division during which the chromosome number reduces to half. For example, if you have started with 46 chromosomes, then at the end of the meiosis, you are going to have only 23 chromosomes in the daughter cells. So the word meiosis was first given by two scientists, Moore and Farmer. This meiosis is a specialized type of cell division which occurs only in the diploid cells. Now, why it is occurring only in the diploid cells? Because here the chromosome number reduces to half. So, there are three features. It occurs only in the diploid cells. The chromosome number reduces to half, and at the end of the meiosis, you are going to get four haploid daughter cells. Whereas, if we talk about mitosis, we are going to get only diploid cells at the end of the mitosis, but in meiosis we are going to get haploid daughter cells. Now, meiosis involves two cycles of nuclear and cell division and only one cycle of DNA replication. Now, what I mean by DNA replication? So, DNA replication simply means the replication of the DNA. And when DNA replication occurs, the chromosome number remains same, but the amount of DNA doubles. For example, if you have 46 chromosomes before DNA replication, then after DNA replication also you are going to have 46 chromosomes. But the amount of DNA doubles. For example, if those 46 chromosomes all together have 12 picogram of DNA, then after DNA replication, those 46 chromosomes all together have 24 picogram of DNA. So you can see here 12 to 24. The amount of DNA doubles, but the chromosome number remains same. Now, this meiosis occurs in two stages. First one is meiosis 1 and the second one is meiosis 2. Now, meiosis 1 is called as the reduction division. Why? Because in meiosis 1, the reduction in the chromosome number occurs. And meiosis 2 is called as the equational division. Equational division means the chromosome number remains same. For example, if I have 46 chromosomes in the starting, then after meiosis 1, I am going to have only 23 chromosomes in my daughter cells. But if I start with 23 chromosomes in the meiosis 2, then at the end of the meiosis 2 as well, I am going to have 23 chromosomes. So, chromosome number reduces in meiosis 1 but remains same in meiosis 2. Now, each meiosis 1 and meiosis 2 has various stages. For example, meiosis 1 has prophase first, then we have metaphase first, then we have anaphase first, and then at the end we have telophase first. Similarly, meiosis 2 also has various substages like prophase 2, metaphase 2, anaphase 2, and telophase 2. So, in the first step, meiosis first occurs in meiosis. So what is meiosis first? Meiosis first is a reduction division. And if we look deep into meiosis first, then in the meiosis first, the first one is the prophase first. This prophase first has various substages like leptotene, zygotene, pachytene, diplotene, and dikinesis. Let us first have an overview of all these stages and then we will learn about all these substages of prophase first with the help of a diagram. So during leptotene, condensation of the chromosome occurs. Now, why condensation occurs? Because in the initial stage of the cell cycle, the chromosomes are present as chromatin fibers, which are type of threads. But for cell cycle, for cell division, it is very important that the chromosome should be condensed. So chromatin fibers are condensed to form chromosomes in the leptotene. Now, in the second stage, that is zygotene, pairing of the homologous chromosome occurs. In the third stage, crossing over between the non-sister chromatids of the homologous chromosome occurs. In the fourth stage, that is diplotene or prophase first, chiasmata is visible. And at the last stage, there is the terminalization of the chiasmata and it occurs during dikinesis. Now let us look into detail of each stage of prophase first. So as you can see here, this is the leptotene stage. So here the chromosomes are entangled. Now these chromosomes are condensed. These chromosomes are condensed. So you can see here, these are the homologous chromosomes. For example, this red one is the chromosome copy from mother and this blue one is the chromosome copy from father. So since these chromosomes are of same type, For example, this is chromosome number 1 from mother, this is the chromosome number 1 from father. So these are called as the homologous chromosomes. Now, during liptoting stage, condensation of the chromosome occurs. Now, during digoting, what happens is the pairing of the homologous chromosome occurs. And during pairing, there is the visualization of the synaptomial complex. You can see here. So, synaptomial complex is visible during zygote. Now, in the third stage, that is patching, there is the crossing over between the non-sister chromatids of the homologous chromosomes. Now you must be wondering what are sister and non-sister chromatids. So if I take this chromosome, then this arm and this arm is what the sister chromatids. But if I'm talking about this arm and this arm, these are what non-sister chromatids. So crossing over occurs between the non-sister chromatids of the homologous chromosomes in the patchity. In the fourth stage, what happens is the chromosomes start separating from each other. The homologous chromosomes start separating from each other except at one side, due to which there is a visualization of the egg-shaped structure. And this egg-shaped structure is called as the chiasmata. And with that, since they are separating from each other, dissolution of the synaptomial complex occurs. So in diplotin, dissolution of the synaptomial complex occurs and chiasmata is visible. Now in the last stage, terminalization of the chiasmata occurs and the nuclear membrane starts fragmenting. So liptotin, condensation of the chromosomes. Zygote, synapses begins, synaptomial complex is formed, bivalents are formed, homologous chromosomes start pairing. Packaging, if I talk about packaging, chromosomes crossing over occurs between the non-sister chromatids of the homologous chromosomes. Diplotin, chiasmata is visible, and in the last stage, dissolution of the chiasmata, or I could say terminalization of the chiasmata. Now, once the dikinesis is done, the cell enters into the metaphase first, where the homologous chromosomes arrange themselves at the equator. They arrange themselves at the equator. Now, in the anaphase first, what happens is the homologous chromosomes start separating, and with telophase, the chromosomes start getting again decondensed. So in the telophase first, the chromosomes again start getting decondensed, nuclear membrane starts reappearing, and we get the two daughter cells. So as you can see here, after prophase, we are having 3 chromosomes in our cell. Okay, we are having 3 chromosomes in our cell. So you can see here, after this meiosis first, we are having only 3 chromosomes in our cell, but initially we have 6 chromosomes in our mother cell. Now these homologous chromosomes have arranged themselves at the equator in the metaphase first, as you can see here. Now the spindle fibers attached to the homologous chromosome. So you can see here, this is the homologous chromosome and then spindle fibers are attached to them. Now anaphase first starts pulling the homologous chromosome towards the poles. So you can see here, the chromosome starts getting towards the poles, and at the end of the telophase first, there is the formation of 2 haploid daughter cells. So we start with 6 chromosomes, but now we have only 3 chromosomes in a daughter cell. And in telophase first also, the cytoplasm starts dividing, and the nuclear membrane starts forming, the organelles starts duplicating, ultimately giving rise to the 2 daughter cells. Now once the telophase is done, there are chances that the cell enters another phase, that is the meiosis 2. But before going into the meiosis 2, a cell needs to prepare itself for meiosis 2 by forming the required proteins and other cyclins and other things. So these proteins are synthesized during interkinases. So what is interkinases? Interkinases is simply a phase between the meiosis 1 and the meiosis 2, where the cell prepares itself for meiosis 2. But kindly note that there is no DNA replication in interkinases, only there is the formation of required proteins for the meiosis 2. Now, once interkinases is done, the cell enters the meiosis 2, and meiosis 2 is similar to mitosis. So, meiosis 2 involves various subphases like prophase 2, metaphase 2, anaphase 2, and telophase 2. And all these phases are very similar to the mitosis. So, in prophase 2, what happens? In prophase 2, nuclear membrane disappears, chromosomes again start condensing. Okay, and this prophase 2 is very small in duration as compared to the prophase first, where crossing over occurs. So no crossing over occurs in prophase 2, and it is small as compared to prophase 1. Now, after prophase 2, there is metaphase 2, where the chromosomes start aligning themselves at the equator, and the spindle fibers attach to the kinetochores of the sister chromatids. So, once the cell enters metaphase 2, then it enters in the anaphase 2. So in the anaphase 2, there is a separation of the sister chromatids. And once the anaphase 2 is over, the cell enters telophase 2, which ultimately gives rise to the 4 haploid daughter cells. So it will be much more clear with the help of this diagram. So at the end of the telophase 1, we are getting 2 daughter cells, which are haploid, each having 3 chromosomes. Now, these 2 daughter cells, when enter is prophase 2, the nuclear membrane disappears, the chromosome again starts condensing. Now, when they enter in metaphase 2, these 3 chromosomes in both the daughter cells align themselves at the equator. The spindle fibers, as you can see here, are attached to the kinetochores, and in the anaphase 2, these spindle fibers pull pull the sister chromatids towards themselves, which ultimately gives rise to 4 daughter cells, 1, 2, 3, 4 at the end of the telophase. So once the 4 daughter cells are formed, we are done with the meiosis. So if I compile both the process, meiosis 1 and meiosis 2, we get this diagram. So initially we have 2 types of chromosomes, each having 2 copies, each having 1 copy from mother and from father. For example, this is type 1 chromosome copy from mother and type 1 chromosome copy from father. Similarly, type 1 chromosome copy from type 2 chromosome copy from mother and type 2 chromosome copy from father. Now, what happens before meiosis? DNA replication. So during DNA replication, you can see the number of chromosomes remain same, but the amount of DNA gets doubled. The amount of DNA gets doubled. You can clearly see this is also a chromosome, this is also a chromosome. But here the amount of DNA gets doubled. Now, during meiosis first, what happens? Reduction in the chromosome number. So here we have 4 chromosomes, but after meiosis 1, each daughter cell is having only 2 chromosomes. Each daughter cell is having only 2 chromosomes. Type 1 copy of mother and type 2 copy of father. Similarly, type 1 copy from father and type 2 copy from mother. Now, after meiosis 2, what we are getting? We are getting 4 haploid daughter cells. So here also the ploidy level is haploid, here also the ploidy level is haploid. But initially we have started with 2n. But after meiosis 1, we have n the ploidy level, and after meiosis 2 as well, we have n ploidy level. So here each daughter cell is having only one type of chromosome, only one copy of one type of chromosome. Okay. Now, what is the significance of meiosis? So meiosis is very important to maintain the chromosome number. For example, if the chromosome numbers are not reduced, then after fertilization or the zygote formation, the ploidy level is going to get 4n. For example, if the gametes are 2n, male gamete is also 2n, female gamete is also 2n, then we combine them, what we are going to get? 4n, which is lethal for the human beings, as it is going to change the ploidy level. So in order to maintain the ploidy level, it is also very important that the chromosome number remains same. And the second reason is it increases the genetic variability. We all know that meiosis have crossing over. So due to the crossing over, there is the genetic variability. If genetic variability is not there, then the chromosomes are passed on as it is from the mother to the father. For example, I get all if for example, if I get all the chromosomes from my mother, then I will be like my mother. There will be no change. But due to this genetic variability, each and every organism is different. And this increases the chances of natural selection. This increases the fitness of an individual. So meiosis is important to increase the genetic variability as well as to maintain the specific number of chromosomes so that the ploidy level is maintained. For example, if the ploidy level is 2n, then after fertilization also it will be 2n. So that is why in gametes the meiosis occurs. So this is all for today. Thank you for joining. Have a very nice day. I hope you all have enjoyed. Bye bye.